Okay, welcome to the session. We'll begin the session now. Thank you all for showing up, being brave enough to come to a pathology session at an oncology meeting. Called the Pathologist Conundrum, and I'll start off by just showing my disclosures. And I would point out that some of the work I'm going to show from my lab is not sponsored by NIH, but rather sponsored by some of the companies below that sponsor research in my lab. What we're going to do today is we're going to have four speakers around the topic of the Pathologist Conundrum. First, Kim Allison will speak about the history of IHC and breast cancer and the ASCO CAP guidelines. Then Reena Philip will give us FDA's perspective on IHC testing as part of drug development. Finally, then Becky Viale will talk about the accuracy of IHC and some studies done, and then we'll transfer over to Andy Beck, who will talk about measuring as opposed to reading, and I'll define that as we go along. So what is the Pathologist Conundrum? So immunohistochemistry tests that are approved as companion diagnostics but have been shown to be non-reproducible in subsequent literature, and there are three examples that I'll go over quickly. But this is the conundrum, and essentially is, should the pathologists be good team players and pretend that it works, or should we insist on changing the assay, or how do we solve that problem of being disingenuous with our client oncologists versus realizing that the literature is discordant with the FDA approvals. So the three examples I'll show is first PD-L1 immune cell staining, and there were three papers on this actually quite a few years ago now in 2017, 2018. But basically, if you look at the, unfortunately I can't circle things here, but if you look at the all assay, if the all assay kappa, FLICE kappa, you can see that it's around 0.2, which is considered minimal, or below minimal, where 0.4 is weak, and 0.1 to 0.2 is considered slight to none in terms of concordance. Here's in gastric cancer, another example in gastric cancer. This was, I was not involved in this. This was done by, led by Rob Anders out of Johns Hopkins. It's a little hard to see, but if I bring up that part, pre-training or post-training, the kappa, the FLICE's kappa was 0.55, which isn't great, isn't awful, but isn't great. And then if we look at a second a study that I ran in gastric cancer, we found the same thing. We look at the OPA, the FLICE kappa, and the ICC, and you can see for CPS 1, less than 1 or greater than 1 on the top line of the table, the OPA is only 31%. So that's pretty lousy, but yet this was FDA approved. And so what's going on? So, but this is a breast conference, so let's talk about HER2 and specifically HER2 low. This is a study that I led on HER2 low, where there was greater than 90% concordance on only 26% of 170 cases read by 17 pathologists. So that's pretty unacceptable for a test that's going to affect a patient's drug prescription or not. That paper in JAMA Oncology didn't allow me to put many figures in, but I think this is the most important figure, which shows that pathologist number 18 thought that only 15% of the cases were 0, whereas pathologist number 1 thought 44% of the cases was 0. So you can see there's a lot of discordance between 0 and 1, but not much discordance between 3 and 2. Remember that for later when I talk about that more in detail. Here's another study. This is a study from Japan that came out in Breast Cancer Research and Treatment. 144 pathologists, 20 cases, an overall agreement of 63.4%. So a little better agreement than we saw. And it may be because they had some training for the pathologists. One of the criticisms of our work is that the pathologists were not trained. They were blinded to the fact that we were going to look at the difference between 0 and 1. When they are trained, this is work that Becky Viale will go into a lot more detail. But over here we see the sort of the high end of concordance at about 80% concordance. So 80% concordance is nice, but how does that compare to other biomarkers? Well, since I also work on lung cancer a little bit, I look at EGFR, and here's the paper that describes a analytic reproducibility test for EGFR mutations. And so you can see that their agreement is 100% every time. Overall, on average, it's greater than 90%, 99%. And I think that's what the patients deserve. We really should be much more precise, not only accurate, but much more precise, and Andy Beck will talk about that a little bit. So how did this get FDA approved? So I hate to reveal this because it's somewhat embarrassing, but the FDA approved this by grouping the 3s with the 0s. And if you group the 3s with the 0s, which makes no biological sense, then you can artificially elevate your approval or your concordance. And so even though they had 93% within reader concordance or 90% between reader concordance, the reason for that was the categories were 0 or 3 versus 1 or 2. But the drug prescription is 0 versus 1. So somehow there was a disconnect, and I don't want to assign blame here, but this is how it happened, that the pathologist conundrum occurs at least for low HER2. Now, what can we do about this? It's not about looking back, it's about looking forward. And looking forward, I think our solution is to switch from reading, which I define as the subjective but expert interpretation of information on a histology slide, to measuring. And measuring is when we do quantitative objective determination of amounts or concentrations, basically an analytic assay. And so you might say, well, that doesn't exist. And in fact, it's just recently exists. This is a paper from 2022 where we did an analytic assay to look at HER2, and we report HER2 in attimoles per square millimeter. And the result, when you compare it to reading, looks like this, where you can see the IHC scores from light to dark, 0 to 3 plus, and then the quantitative scores with the analytic assay comparing, you can see that there's some pretty dark blues over on the left side of the scale, which suggests that they're not even below the limit of detection, or limit of quantification rather. There's a couple of cases that are 2 plus unamplified, suggesting this is a problem that it's not that pathologists are not good at reading, it's just that human beings can't tell subtle distinctions. And I think that's why we need to move from reading to measuring. Does it make a difference? Well, this is actually work that was presented at ASCO by Palo. And for every 5 attimoles per square millimeter increase, there's a hazard ratio of 0.7, which is statistically significant, which means the more you have, the better the patients do. And so this is sort of the justification for reading, which we've seen sort of when you compare between trials, you can see sometimes subtle justifications for reading. And we know that, for example, the HER2 amplifieds in the trials for TDXD did much better than the HER2 lows, which in the DAISY trial did better than the HER2 zeros. So there's a clear, there's a suggestion in the literature that there's a proportionality to amount, but this is the first proof, and it's not yet published, but it was presented at ESMO, that it's actually continuously across the scale of expression. And the top is the HER2 amplifieds, and the bottom is the HER2 unamplifieds. Very small study, it's the RELIEVE trial, then we're expanding that now. And so not to say that we're the only people that can do quantitative work. This recently appeared from AstraZeneca, where they do HER2 continuous reading. And I won't go into this too much because I'm going to have Andy Beck really talk a lot about continuous reading. So without further ado, I want to now introduce each of the speakers. There's 2 options, so we're going to not have questions after the speakers. We're going to have questions all at the end. And if we all keep our time as best we can, we should have about 15 to 20 minutes for questions. There's 2 microphones in the room, and we'll take questions from the microphones first. However, I will also take questions if you want to put them in with your phone. I can see them up here on the monitor, and I can take questions that way as well. So without further ado, Dr. Allison from Stanford is going to give us a brief history of the IHC HER2 story and ASCO CAP guidelines.
Thank you, Dave. Good afternoon. Thank you, David, for the invitation, and thank you for the organizers for having a topic that is pathology related and talks about the conundrums that we face. So I'm going to be talking about the history of IHC and breast cancer. Let's see if we can get it. No.
There we go.
There we go. It'll be a brief history. I don't have any disclosures. So I'll spend the next 15 minutes giving some highlights of the history of IHC markers. I am going to focus on HER2. I'm going to review some of the evolution of our guidelines in breast cancer and IHC testing, again, focusing on HER2, and talk about some of these strengths and limitations that have been raised in predictive biomarker testing by immunohistochemistry, again, emphasizing how low can we go in HER2, because that's a topic we presently have a conundrum with. So our story really begins with ER testing, and this was the first shift away from the grind and bind assay, which was ligand binding assay at the time, to immunohistochemistry to identify ER positive cancers that might respond to hormone therapy. And this was a positive thing because we had a need for a large amount of tissue, fresh frozen tissue with radioactive agents. And these assays, while quantitative, giving us a nice quantitative discrete answer, really admixed normal and in situ lesions. So those advantages, the advantages of IHC really were that you could see what you were scoring in situ. Pathology labs had access to this technology. It was low cost. You had fast turnaround. We can do it in formal and fixed tissue. We can see small invasive cancers and score them in situ. So a visual scoring method key. But early days, scoring methods and thresholds were really not well standardized. Labs would use whatever threshold they decided to. For ER, for example, there was not a standard. And there were various antibodies and methods with variable sensitivity, including differences in analytic issues like antigen retrieval. And then, of course, pathologists' interpretation, which we'll be talking about here, does require education. And in the early days of HER2 IHC testing, this was a big shift because it was a unique scoring system that hadn't really existed before. We were used to any staining meant something was positive versus negative for diagnostic purposes. And with HER2, we were really looking for the overexpressed, so intensity of staining mattered. We weren't used to scoring that. But luckily, we also had a reflex test we could use to help us out when we had equivocal or 2+ results where we couldn't tell if the intensity was truly due to overexpression due to gene amplification. So we had FISH testing. And really in the early days, it was just a marker of aggressive biology. So we were doing it on at some labs to indicate who had a poor prognosis and might need more treatment. And early biomarker guidelines in the 90s, really only recommended ER and PR testing. They didn't even recommend a specific threshold at the time. And they did not recommend HER2 testing, and even went further and said, we don't think it ever will be. So times have clearly changed. And with the metastatic trials and then the early breast cancer trials with trastuzumab, of course, HER2 became a high-stakes test. It was now a predictive biomarker linked to treatment. And then came the calls to standardize this test. We really had to have high-quality results for our patients, and there were concerns that up to in those Herceptin trials that up to 20% of the local labs submitting to the central lab as a HER2 positive couldn't be confirmed, so false positives. So Dr. Antonio Wolf with ASCO, who's here with us today and was speaking on this topic earlier in this meeting, and Dr. Elizabeth Hammond, our pathologist with CAP, joined forces and said, hey, let's get both of these organizations together. Let's really set some standards. And that has made a huge difference in HER2 testing. First guideline was published in 2007. Here's the list. I won't read them all. I'm sure many of these names are familiar to you. That really helped us gain expertise and what we need to do to make this test reproducible and valid for our patients. The suggestions and recommendations in there really cast a wide array from like pre-analytic factors to how to handle tissue, to scoring and reporting, and then also importantly included validation, QA, external proficiency testing, all tied to lab accreditation, right? That made it something enforceable. All of this was really designed around reducing HER2 testing variability, focused on discriminating the positives, overexpressed, amplified from not. And the table 3 from that initial guideline shows the sources of HER2 testing variation that can impact our results. Pre-analytic issues like time to fixation, type of tissue processing you're doing can risk a false negative result. Again, we're focused on the positive versus the negative, not the subtleties of the ultra low versus 0 at that time, but they're certainly sensitive to that. And then post-analytic issues, this was the interpretation side, and the issue was false positives with over-interpretation of a 2+ as a 3+. So those guidelines set the standard initially in 2007. They've iteratively changed with focused updates in 2013 and 2018, fine-tuning, getting rid of equivocal categories for FISH, addressing less common scenarios, all again with the goal of accurately identifying our overexpressed amplified from not. And these guidelines did result in improved standards. We are actually quite good now at determining positive versus negative. This was the scheme that has stood the test of time, again, to emphasize how we determine positive versus negative in terms of overexpression or amplification. There are subtleties to HER2 IHC interpretation that pathologists do need education on. There's unusual staining patterns, things that can't all be detailed and covered in a guideline, including heterogeneity, and then importantly, checks the pathologist can correlate with what they're seeing. If it's a grade 1 low-grade cancer and I'm seeing a lot of HER2 expression, hold on, is something wrong here? I need to correlate with my histology. So I think that's a really important role that pathologists play, regardless of what result comes back from where, is we have to correlate it with what we're seeing as the biology. Our FISH testing is also not necessarily not going to have gray zones like the IHC testing does. And we, in 2018, addressed some unusual combinations of results where we don't have as much data on outcomes and response rates for different combinations of signals per cell and ratio. So the group 2, 3, and 4 results were created with gray zones and borderline results. I think the message is that we want to confirm them. We do additional counts. We want to correlate with IHC in this case, and we want to explain. And we had a bunch of recommended comments to explain this is a gray zone, this is an unusual result. And I think those have been useful over time. And then last, importantly, proficiency testing in QA programs have, the majority of labs now participate in one of these programs across the globe. There's College of American Pathologists, there's also other programs around the world that are really helping us to ensure this is high quality testing. So I think that is good news. This is the good news, right? It's been predictive. This HER2 test, the same test, across an expanded menu of targeted treatments. We don't have different antibodies that we have to use for different indications like we do in PD-L1. One test, and we've predicted all of these different therapies that have expanded in this menu, including the newer antibody drug conjugates and TDXD. Game changer, right? Destiny Breast 04. Now we have an antibody drug conjugate that works in HER2 negative, or now HER2 low was the term. And this was great news. Could use it in the metastatic setting, but really what are the HER2 testing implications is what we're going to talk about. We know our amplified overexpressed. It's predictive. When we get those test results, DB04 really shifted the bar down so that our 1+ and 2+-ish negative, these patients, because of the way the trial eligibility criteria worked, were eligible for the trial, which was positive, so now they're eligible for this therapy. The IHC zeros were excluded, so we didn't know if they responded to TDXD. We now have a little more information on that group, but not complete. So we kind of had an artificial threshold reached between 0 and 1+ that pathologists now need to focus on discriminating, even though we don't really have a validated new predictive threshold. FDA approved the same test, the 4B5 pathway antibody for HER2, but now with a new indication, just expanding it. And there were lots of questions for the guidelines. Shouldn't we be changing our terminology? Should our reports now say HER2 low? There were a number of problems with the HER2 low terminology. And first off, the trial used the ASCO CAP current IHC scoring criteria, 0, 1+, 2+. So we really had that information in our pathology reports, didn't need to change our scoring, used the same scoring system. So that was great news. And there really wasn't a new predictive or prognostically relevant category. It was more trial eligibility criteria, in addition to other practical issues with if you have an IHC 2+ result, you can't necessarily call it low until you've got your FISH result back, right? So all these issues made it challenging to adopt a new terminology. And then really the concern is that IHC may not be fit for the purpose of detecting these lower levels of expression. And work that David and others have done have shown that IHC 0 may not really mean no protein, it means no protein detected by your assay, and they may actually also be HER2 low or ultra low, looking at more sensitive methods like AquaIF. So this is our new conundrum for HER2. We've got a new clinically relevant, although untested threshold. There's issues with reproducibility of that threshold, and it's not what the assay was designed for, and it's likely very related. The IHC 0 is related to a limit of detection of the assay, fixation issues, very sensitive to pre-analytic factors. We don't have another reflex assay yet to help us discriminate if we are on the fence, and it was unclear how to standardize it to that very low level. So our update for 2023 was really just an affirmative update. We did it as fast as possible, basically saying we're not changing anything. There's no changes to reporting, but we have best practices we can recommend, and we really need to spread awareness to pathologists so that they are aware that those lower levels scoring make a difference. You need to look hard. You need to look on high power. Use a second pathologist as your additional stain, I guess, to if you're at a threshold. And then use controls with a range of expression. And I think we've still got work to do to make sure we're accurate at that low end. And then because it appears that HER2 low shifts from sample to sample in the same patient, from primary to metastatic and back again in different metastatic sites, can be 0 versus non-zero, it didn't look like it was a biologically reproducible phenomena. It may just be assay. Your one sample's fixed better than the other, or the electricity running to your IHC machine was stronger one day. I mean, this is a very subtle difference. So using any sample was another recommendation when making these decisions. So giving patients options and eligibility. Again, of course, now Destiny Breast 06, since it's the spring, has now expanded that. Their population intent to treat included part of our 0 category that had any staining, less than 10%. It moved the indication to an option earlier in treatment, and that was really the main endpoint of that study, but it expanded the eligibility population to this additional group where any staining could matter. It really, wasn't powered to focus on this ultra-low population. There were only 76 in each arm of this ultra-low group, 76 patients. And you can see these curves do cross and that it was not statistically significant, so it wasn't really powered for that. But now, because they were included and it was a positive trial, we're being asked to split the zeros and determine no staining from faint incomplete membrane staining in less than 10% of the cells. And just to show you an example of how subtle this is, on the right we've got a no staining, and this is from the KIT interpretation guide, to less than 10%, maybe that's a whiff, so that's an ultra low, and then a slight bit more, and you've got a 1 plus, a true greater than 10%, so extremely subtle. Despite these concerns, we need to include this information in our reports so that we can figure out if a patient might be eligible. And the good news is we don't need to change our criteria. Our scoring criteria, again, the same were used, but we need to unlump this IHC 0 category for reporting purposes. If we tried to update guidelines, this would take a year or two, gather an expert group and have a bunch of meetings. It's really a reporting issue, and so my approach has been to work with CAP, the College of American Pathologists, to work on our breast cancer biomarker reporting templates to try to make sure that that standardized template at least has the information that is needed, separating out the IHC 0 to both categories that are included so we can get specific. I like to call the ultra low 0 plus. You could argue that maybe we shouldn't even distinguish between the two, and you could just say no staining versus some staining, but again, that would require a whole new schema and reporting strategy and guideline revision and evidence behind it that it works. So again, problem of controls and validation. How are we going to do that at this really low, subtle level of staining? We don't have controls that we can say every day are reliably going to be the same. There's probably heterogeneity. CAP is not planning to do proficiency testing for ultra low and 0. They are planning to try some image interpretation And the reason for that is because if you're sending a TMA to multiple labs, with thousands of labs across the world, that each level you would have to validate is ultra low versus 0, and any level you do, it probably can change, right? These are heterogeneous little whiffs of staining. So, I think the final question is all a moot point? With the DAISY trial, I'm sure you're all familiar with, there was response in that IHC 0 group. Is this because of heterogeneity? Were these ultra low? Or I think most likely it's a false negative IHC 0, and this may respond. We may have a much larger, even larger group of patients that will respond. And I love this diagram. I think, the times keep changing, and we may go from HER2 positive to negative to now we've got these low and ultra low categories that seem to make a difference in terms of eligibility, but not validated thresholds. And maybe we're going back to where we started, actually. Take-home points. So well-standardized assay, we really do a good job of overexpressed amplified versus not. Lower levels of expression are a concern. It's a limit of detection issue, in my opinion. We need to test those predictive thresholds better in trials, not just to use them as eligibility. And we need innovation in quantitative measurement and scoring if it matters. We need validated standards to measure against. And I think on my wish list, it would be great, now that we've got so many more options in the metastatic setting, how do you prioritize them? How do you decide what to try first? Wouldn't it be great if we had biomarkers that actually could tell you a little bit more about risks, like risk of interstitial lung disease? Is there a biomarker we could try? Anyway, that's my wish list. That's all I've got for you.
Good afternoon. Thanks for inviting me to participate in this section to talk about FDA's perspectives on IHC testing in breast cancer drug development. I have no financial relationships to disclose. So I'll start with providing some background regarding the trends of precision oncology drug approvals, followed by the different biomarker-based trial designs used in oncology drug development. And then I'll cover details of companion diagnostics test validation with emphasis on IHC testing. And we'll also provide some future considerations when using IHC assays in drug development. So as we all know, in oncology over the last 20, 30 years, there has been an increase in biomarker-targeted drug development programs and an expansion in the number of FDA-approved cancer drugs that are effective in biomarker-defined populations, starting with approval of Herceptin for the treatment of HER2-positive metastatic breast cancer. And depending on the clinical trial design, there has been an expansion in the use of predictive or prognostic or selection biomarker strategy in clinical trials. These biomarkers they are used to identify individuals who are more likely to experience a favorable or unfavorable effect from a therapeutic intervention. And the ability of the biomarker to accurately classify patients, it depends on the cut points and thresholds, and that could very well be different by different tumor types. So the analytical validation at around these cut points are as important as clinical validation when these biomarkers are used in precision oncology. And we will get into the details of analytical and clinical validation later in my presentation. So whether IHC or any other methodology is used to select or stratify patients in clinical trials, the two most commonly used clinical trial designs are either biomarker positive only trial design or biomarker stratified design. Biomarker positive trial design, it's also called as selection trials. This trial design is recommended only when there is compelling evidence that the drug works only in marker-positive patients. So in this trial design, biomarker selects patients for the study, and only marker positives are included, marker negatives are excluded from the study. Most companion diagnostics are selection biomarkers. That is, the trial was a biomarker positive trial. And this trial design is also good for selecting high-risk populations So there is an increased event rate and therefore a smaller sample size needed and leads to a shorter trial. The second trial design is the biomarker stratified trial design. Patients first have biomarker assessments, then patients are then stratified into separate cohorts based on the biomarker status, then randomized to the experimental or the control arm. And ideally, the therapy is tested in each biomarker population individually based on an adequately powered pre-specified statistical plan. When it's a biomarker stratified design, depending on the clinical trial results, there are three possible outcomes. When patients benefit, regardless of their biomarker status, the agent would be approved in the all-comers population and would be labeled without describing marker positive or marker negative results. However, if there is a differential effect, but the biomarker negative patients are still felt to have a favorable benefit-risk assessment, the indication would be approved for all comers with a complementary diagnostic, and Section 14 of the drug label would include details of the efficacy by biomarker status. In instances where there is a differential effect and the biomarker negative patients have an unfavorable benefit-risk assessment, the agent would be approved in the biomarker positive population with the companion diagnostics. Most of you probably know the definitions of companion diagnostic and complementary diagnostics. However, I'm showing those definitions here for your reference. Definitions of complementary diagnostic is still in a draft form since there's no guidance published for complementary diagnostic. There are only a couple of such tests approved so far, although the concept makes a lot of sense because it helps a doctor to make judgments about the usage of the drug, but there's not a lot of incentives for sponsors to develop complementary diagnostics. As we discussed in the previous slide, the decision whether it's a complementary or a companion diagnostic, it depends both on the design and outcome of the clinical trial. So the link in this slide, it takes you to the list of FDA cleared or approved CDX. IVD, in vitro diagnostic tests, could go through either a 510K pathway for moderate risk devices, and they are called cleared devices. Or IVD tests could go through a PMA pathway for high-risk devices, and they are called approved devices. So most CDX in oncology have gone through PMA pathway, and therefore they are approved devices. FDA's decision summaries for 510K cleared devices and SSED, summary of safety and effectiveness data for all PMA-approved devices are publicly available. These include the details of analytical and clinical validation studies performed using the tests that supports the safety and effectiveness of the device for the specific indications for use. So generally, FDA review of IVD companion diagnostic device regulatory application is conducted contemporaneously with the review of the associated therapeutic product through a collaborative interaction between the different centers, CDRH, CDER, and CBER, depending on whether it's an NDA or BLA. The review of these regulatory applications include the assessment of the device's pre-analytical, analytical, and clinical performance provided by the sponsor. Please note that this is the data that's provided by the sponsor that will be reviewed by the agency. This is to determine the safety and effectiveness of the IVD companion diagnostic when used with the corresponding therapeutic product. Next few slides, I'll cover these in detail. As we will cover later, validation studies of the CDX test should be performed using the final to-be-marketed test with the final protocol and should use specimens from the intended use population with specific attention to the assessment at the pre-specified cutoff points. As said in the previous slide, FDA review of IHC assays include assessment of pre-analytical performance, pre-analytical variables that can impact device performance. Some of the pre-analytical variables include the specimen type, fixation type, tissue pre-processing, tissue thickness, etc. Robustness studies are also needed. These studies are conducted to assess the tolerance in minor variations in testing conditions, such as tissue thickness, antigen retrieval, pH, temperature, time, antibody concentration, incubation time, etc. A locked protocol should be used for all steps of all the analytical and clinical validation studies. As said earlier, FDA review of the IHC assays include analytical validation studies. Analytical performance is the ability of the test to accurately and reliably measure the analyte of interest, and it is supported by these studies. Analytical sensitivity study, it is a prevalent study in the intended use population. Analytical specificity study is characterization of the antibody, which include 3 different studies: Western blot, peptide inhibition, and tumor body and tumor study. And precision of the assay is evaluated with 3 different studies: reader precision studies, intermediate precision study, and interlaboratory precision study. Intermediate precision study includes studying precision of different lots of antibodies, instruments, detection kits, days, etc. And the marketed tests assess the site-to-site reproducibility in at least 3 sites. Since IHC tests are assessed manually by pathologists, reader reproducibility measures are an essential part of the performance characterization. Mainly 3 stability studies are conducted: cut slide, real time, and ship stress stability study. These studies inform stability instructions for the assay. As indicated earlier, the indications for use, or in other words, the type of claim of CDX, depends on the clinical trial design and outcome, and results from a successful trial provides the clinical validation of the CDX. So when biomarkers are incorporated into clinical trials, all clinical sites should follow the same protocol for sample collection, processing, and testing. And clinical trials used for patient enrollment, the CTAs used for patient enrollment, they should be fully specified, cutoffs established and locked down, and there should be a uniform and consistent biomarker definition. CTA should be analytically validated and should have minimum performance characteristics. So ideally, final to be marketed tests should be used as CTA in the pivotal trial. However, most often, final to be marketed test is not used in the clinical trial. That is, the clinical trial used for enrollment is different. Clinical trial assay that is used for enrollment is different than the to be marketed CDX. So then a bridging study should be conducted for clinical validation of the CDX. Therefore, pharma should have a robust plan for specimen banking and retesting. In a marker-positive trial, it should make every effort to bank all marker-positive samples and sufficient representative marker-negative samples for a bridging study for retesting with the final to be marketed CDX assay. And sensitivity analysis should also be performed to demonstrate efficacy of the therapy is preserved with the CDX. So in this conference, we have heard a lot about the tissue-agnostic drug development. That is, a drug is developed for an indication defined by a biomarker across multiple cancer types. So Enhertu approval last April for all solid tumors that are HER2 3+ positive is an example. FDA has published a guidance on this, and some key points are included here. When anyone is considering the tissue-agnostic approach, should consider that the tissue-agnostic biomarkers should be supported by a strong underlying biological and clinical rationale. There should be data supporting that the biomarker presence indicate similar biology across different histologies. Preliminary studies should be done to determine assay cutoff is the same across different tumors. In addition, clinical trials should explore efficacy and demonstrate efficacy across multiple tumor types. Also, the assay performance should be demonstrated across different tumor types, especially across tumors that are prevalent and those that are enrolled into the trial. In addition to analytical and clinical validation, one of the most important aspects is the alignment of biomarker rules. The sponsor should ensure that the biomarker calling rules to define a biomarker is consistent between the enrolling CTA assays, and if there is a different assay that's coming in as a CDX, then it should be consistent with the final to be marketed CDX assay. Wrapping up my presentation with some future considerations on the clinical as well as the analytical aspects. As discussed earlier, when mechanism of action, non-clinical data, prior clinical studies suggest a differential efficacy based on the presence or absence of a biomarker, pivotal clinical trials should be designed to provide an adequate benefit-risk assessment for both biomarker positive, biomarker negative population. In such situations, appropriate trial design to evaluate the effect in the biomarker negative population is needed. This includes randomization that is stratified by biomarker status and an adequate sample size accrued to the biomarker negative population to obtain a precise estimate of the treatment effect. Appropriate trial conduct also requires a complete ascertainment of biomarker status. Moving on to analytical aspects, as we heard earlier, IHC is an extremely valuable clinical test for assessment of protein biomarker expression in tissue. With that said, multiple manual steps in the process is making the reproducibility difficult, and differences in scoring algorithm between different sponsors is making the interchangeability difficult. So there are a few points for consideration, as mentioned by the previous speaker. The need for uniform biomarker definition, scoring methodology, criteria, scoring algorithm for each indication. This should be consistent between assays, and when a new biomarker is introduced with IHC testing methodology, harmonization efforts are needed to improve consistency between assays in clinical settings. For some IHC-based biomarkers, uncertainty surrounding cut points has led to numerous trials in which treatment effects are assessed within subgroups defined by more than one cut point. When multiple cutoffs are assessed in a trial, analytical validation should be done at around each cutoff before drug efficacy is evaluated. There is also a need for development of objective scoring methods. Validated image analysis softwares may help. We'll hear about that in the later talk. It may address subjective assessment of qualitative output. There is a need for reference standard for assessing analytical accuracy. There's a need for development of analytical standards, and that could mean in more robust analytical validation. And this could also address some limitations of IHC assays we heard, such as the small dynamic range and difficult to quantitate. Lastly, I want to acknowledge those who have supported me with the content of this presentation. Thank you.
Okay, next Dr. Viale will give us a view from the proficiency testing world.
Yes, David. First of all, thank you for the opportunity of participating in this session, despite the fact that I will have to address some critical points. These are my disclosures. So we will be talking about accuracy of immunohistochemical testing. In general, we know that accuracy may depend on several different aspects. We have pre-analytical variables, like promptness, type, and length of fixation. We have analytical variables, sensitivity and specificity of the assays, the staining protocol that has been used, the performance of the immunostaining platforms. And finally, we have issues with the interpretation and reporting of the immunohistochemical results. Depending on the knowledge and expertise of the reader, the scoring system that has been utilized and the consistency of the report. I will try and focus on the HER2 low breast cancer. We know that the patients that have been enrolled in the DB04 and DB06 studies have been enrolled by central assessment of HER2 low status using the Ventana HER2 4B5 immunohistochemical assay and the ASCO CAP scoring system. Then the drug has been approved with regard to the population of the DESTINY-Breast04 study, and it has received a breakthrough therapy designation for the population of patients in the DB06 study, together with the companion diagnostic labeling of the pathway 4B5 immunohistochemical assay. So this is the starting point. What we know today is based on these factors. So how can we evaluate the accuracy of immunohistochemical staining, specifically for this HER2 low / ultra low expression? Well, theoretically, we have several options. Maybe the most common three options are performing ring studies. And these studies will help us to evaluate the interpretative aspects, because in general, these studies will provide the participants with either the actual immunostain slides or digital images of the immunostain slides. So it's not an issue of controlling pre-analytical or analytical, it's just controlling interpretation. Then we have these external quality control schemes. They will help us to evaluate both the analytical and the interpretative aspects, because many of these schemes, they will send unstained tissue slice, and so the participants are expected to stain and read the immunostain slice. I'm more confident with some European external quality control schemes like the UK NEQUAS and the Nordic CQ, and I will briefly touch upon them. And finally, maybe most interesting, are the results of the real-world data. What's happening currently in the world when we have talking about accuracy? Theoretically, this real-world data will enable us to evaluate pre-analytical, analytical, and interpretative aspects. As a proxy of real-world data, I will discuss briefly what has happened when local evaluation of HER2 low and ultra-low status has been performed in the conduct of these DB04 and 6 clinical trials, and the comparison between the local versus the central assessment. So let's start with the ring studies. David already has pointed out, we had very alarming results, and this has been already discussed. Certainly, the participants in the study were not trained on the low expression of HER2, so possibly this could have been a little bit improved, the results, if participants would have been trained. But after the first alarming results, there have been more and more ring studies all over the world. These different ring studies have a variable number of samples. Some are very small, some are quite large, with a variable number of participants. And this is the important aspect, variable interpretation of concordance. So some of these ring studies have just compared the results of the participants according to the 4 scores, 0, 1 plus, 2 plus, 3 plus. Others have put 0 versus all the rest. And more recently, with the ultra-low status, some of these ring studies have compared the immunohistochemical 0 score without staining versus all the rest. So it is very difficult to derive from these different studies what is the status, the current status of the accuracy of immunohistochemical staining for this low expression of HER2. Certainly there are so many studies, and I have been involved in one of them. And this is a global ring studies that we have conducted. There were 77 pathologists from 14 countries. All of them have received a 4-hour virtual training session. And we have sent them 50 cases of breast cancers with different scoring results. Immunostained with 4B5 or immunostained with the HERCEP test according to the assay that was used in the different sites. So this is a table showing that in general we have approached a positive % agreement and a negative % agreement, which was higher as we were expecting. And a second point is that after training, there was not a very significant improvement, but there was still some improvement, especially because we achieved a reduction on the false positive scoring and an increase in a negative % agreement, telling us that we were more consistent in the identification of HER2 low following the 4 hours training session. So there was some improvement. Let's now go shortly to the external quality control schemes. This was, as far as I remember, the first run of the UK NEQUAS for including HER2 low disease. And you see here in the results that the performance of the 36 participant labs was quite poor when the cases were scored 1+, and this is the sample number 1 and number 2, whereas there was an improvement in the agreement among the different labs when it came to immunohistochemical score 0 or 2+. So again, focusing on the difficulties of the pathologist in properly identifying those tumors with low expression of HER2. This is quite similar what has been obtained by the Nordic QC. This is a more recent study. Is a run on the, it was run in 2024. And you see in the bottom of the figure that even in case of low expression, case 1 and case 2 and case 3, you see that they were have achieved an agreement among 326 labs that was approximately around 70 to 75%. It's not very good, it's not very bad, it's something that could be a good starting point for improvement. And then I will finish with this proxy to real-world data. This is a comparison within the DB04 study of local versus central scoring of the samples that were submitted for patients that could be eligible for the enrollment in the trial. We had 160 sites from 19 countries. And the bottom point is that, again, around 75, 78% of those samples that were locally scored as HER2 low actually were confirmed as HER2 low by central testing. So again, we are between 70 and 80%. Let's go to the more recent comparison. This is within the population of patients in the DESTINY-Breast06 trial, again, central versus local, 324 sites from 28 countries, and again, the overall % agreement for HER2 low status was around 75 to 80%. So this seems to me to be very consistent in all these different data sets. It was surprising when I presented this data in Barcelona that they say, okay, but if you look at the local score 0, you see that there was plenty of those cases that were not actually, that had some staining, and so pathologists are not able to. You have to remember that in the DESTINY-Breast06 trial, local pathologists were not supposed to identify within the 0 category, the 0 score, those cases with some staining and those cases without staining. So this is not a fair comparison. Let's focus on those cases that were local and centrally confirmed as HER2 low. And this is, again, approximately 80%. This is something that we have added. You know that both DB04 study and DB06 study, some 20 to 30% of the patients had not sent for central evaluation the biopsy of the metastasis, but samples of the primary tumor. And so the question arose whether all the patients to be enrolled or to be candidate for the treatment with TDXD should be tested on a fresh biopsy of the metastatic sites, or if we could trust on the results of the immunohistochemical stain performed 3, 4, 5 years ago on the primary tumor. And so this was a study that we have performed. We have taken some 800 historical samples that were patients with metastatic disease that were diagnosed, and the tumor was immunostained in the years between 2014, 2017, and they were considered those days as HER2 negative. Those days there was no concept of HER2 low or ultra low, so it was just or 0 or 1 plus or 2 plus without any amplification of the gene. We have rescored, not restained. We have just retrieved those immunostained slides, and we have rescored today with the concept of HER2 low and ultra low in our mind. And again, you see here that when we looked at the cases that historically were already considered HER2 low because it was 1 plus or 2 plus without gene amplification, in 80% of the cases, again, we could confirm HER2 low status upon rescoring with the today concepts in our mind. But those tumors that we had considered and scored as HER2 zero in the past, when reviewed today, some 30% of these tumors that were considered HER2 zero, now we will consider HER2 low. So the final message is that within the multidisciplinary discussion, certainly the oncologists may ask us to rescore the past samples, because if it was already HER2 low, we think it is trustable. But if it is zero, we have to rescore it, and most likely 1/3 of these patients actually will be rescored as having a HER2 low disease. This is my epilogue. So certainly accuracy and reproducibility of routine immunohistochemical testing for HER2 low breast cancer remain less than satisfactory. In the best possible scenarios, after education of the pathologist and training, concordance among readers is still around 80%, not much more than that. I'm pretty sure alternative approaches may well be more precise and more reproducible in the measurement of HER2 expression in breast cancer. But there is something that I'm always concerned a bit about. We are running a lot of this analytical comparison, looking for sensitivity, what is most sensitive, less sensitive. I would like to say I'm not that much fond of sensitivity. What we need is prediction. And I'm not sure that a more sensitive test will invariably translate into a more predictive test. So this is the reason why those bridging studies, to me, are very important to be run. Thank you very much for your attention.
Okay. Next, Dr. Andy Beck is going to take us through quantitative approaches to HER2 measurement. But in the meantime, those questions that you've been thinking about in your mind, you can put them in to the on the app and put them in so that we can begin with your questions when Andy is finished.
Great. Thank you so much to the organizers for this wonderful session and for including me in it. And I'm excited to go last because I do feel like AI can really significantly assist for a lot of the exact problems we're seeing. Oops, sorry. My only disclosure is I'm an employee and equity holder in PathAI. And just to restate the conundrum, it is that pathologists are extremely good at their job. They have incredible judgment. They cover thousands of different diagnoses and have to express a lot of clinical judgment. But there are some new CDXs on the market. We talked about HER2. I'd say PD-L1 is another, quantitative assessment of Ki-67. And there will only be more in the coming years where they're just being asked to do an essentially, in my opinion, impossible task. if you just picture that every single slide has on the order of hundreds of thousands of cells, and as you saw even in Kim's example of negative versus low versus high on one tiny little field of view, that probably represents 1% of one slide, and they may have to go through hundreds of slides in a day. There's just not enough time to actually apply the criteria that are being asked to apply. And I actually do think pathologists could apply it if they were just looking at small fields of view. We've seen this many times. But the millions, hundreds of millions of cells that you have to look at each day is literally just impossible for a human. It's not impossible for a machine to do that. And an issue with things that require expertise and a lot of human judgment that we often miss is that wherever there is judgment among humans or even within a human, there is noise. And we focus a lot on accuracy, which is important. And that's sort of an accurate and reproducible test, or an accurate and precise test is indicated there in the top left, where essentially the test is producing a result right in the center of the bullseye, and it's doing that essentially every time. But even if a test is accurate on average, if it's noisy, many patients will have diagnoses or interpretation of biomarkers that may not be correct for getting them on the right therapy. And that's kind of indicated here on the top right, where you have a test that on average is hitting the bullseye. But if you look at every individual, there's a huge dispersion. So anything that requires a lot of judgment, or it's a very difficult task, or has a lot of the challenges we're talking about, is going to have this imprecision, and we've heard about that throughout the talks today. So this is something where algorithms do extremely well. So if they can be made as accurate as a consensus of experts, they are by definition going to be far more reproducible. And this is just a quote from Danny Kahneman, where he says, "We know that in many situations, algorithms do better than people in the quality of their judgments because they are noise-free," meaning that given the same inputs, they will produce the same outputs. And this is an extremely important observation for both clinical trials, but also for clinical practice. And just to give you a couple examples of this in action that I think is powerful. So this is a study we did on AI-based scoring of PD-L1, and the main purpose of the study was showing how reproducible the predictive measurements were, which were also very reproducible. But this is just showing the analytic reproducibility, where here we have 6 slides ranging from totally negative in PD-L1, which would be slide 5, to over 75%, slide 2. And it's been scanned twice, and the same algorithm's been applied twice. And you get essentially exactly the same readout for the same patient. So if you're one of those patients, and an algorithm is being applied, you can be sure you'll have essentially the exact same readout. And these AI systems are globally deployable. So whether it's seen in any lab, doesn't actually matter which pathologist is necessarily operating the machine, you will get the same result. And that's just, as we know, absolutely not true for manual scoring. Even the same person often will give different quantitative estimates when looking at the same slide on different days, let alone variability among people. So that's an example from PD-L1. I'm just going to show a couple examples from scoring of liver disease, which is not obviously breast cancer, but has many of the exact same challenges. This is a study we published earlier this year in Nature Medicine on an AI-based system for scoring of liver biopsies, which is extremely difficult for humans to do. There's tons of lack of reproducibility. And the journal editors just asked us, and we thought it was kind of trivial, but why not do it? Because I think it actually is powerful data. If you just run it twice, does it agree? Because they've seen so often a human, you show them the same image twice, you get the same answer something like 70% of the time. Here we just, for all the 630 plus slides, ran the same model multiple times, and we got 100% agreement. So just like that, knowing that you're taking noise out of the equation and you can focus only on accuracy is very, very powerful, because noise probably contributes as much to error as accuracy does. That's kind of what Danny Kahneman's book argues. And just to give you some example of liver, this also reminds me of Kim's example in HER2 breast, but it's just an impossible task that's being asked to do. One of the key criteria for both getting a diagnosis of non-alcoholic steatohepatitis is you have to have ballooned hepatocytes. And then one of the criteria for determining whether a treatment is effective is, has NASH been resolved, and has the number of ballooning hepatocytes gone down? So you get a biopsy before treatment or biopsy after treatment. And in this study, they just asked a group of 9 expert liver pathologists exhaustively hand label every ballooning hepatocytes on about a dozen slides, and they had about 10 pathologists. So overall, they had tens of thousands of individual labels. And out of all of the tens of thousands of cells that were labeled, only a single hepatocyte was canonically agreed to by everyone. So this in the whole study was the one cell that everyone agreed is really a canonical ballooned hepatocyte. And the job of the pathologist is actually to count each of these across every biopsy. So it's just really not possible to apply that criteria, which is so critical for approving new therapies in liver disease. And the Fleiss-Kappa statistic was about 0.2 in this study. And they concluded here that the substantial divergence in hepatocyte ballooning identified among experts suggests that ballooning is a spectrum, too subjective for its presence or complete absence to be unequivocally determined as a trial endpoint. So this really motivated us for another study we just published earlier this year in Nature Medicine, which was on the clinical validation of our AI-based tool. How can we build a system that would combine the AI and use that to assist pathologists and then assess whether it improves agreement with the consensus ground truth of experts? So here the ground truth is still the standard of care ground truth, the ground truth that's used in clinical trials. It's a consensus of humans not using AI. But the experimental arm here indicated on the left is we have pathologists review the AI output and either agree with it, or if they think it's far off, they disagree with it. And then we can compare two trial arms, independent manual reads versus pathologists augmented by AI. And this is kind of one of the themes of the study. I really think this will be the future paradigm for how AI systems should be clinically validated. They're not just algorithms in their own. It's not just analytic validation. It really is, how does this impact physician behavior? And here we showed significant improvements of pathologists augmented by AI for agreeing with the manual scoring ground truth, particularly in lobular inflammation and ballooning, which are two of the key factors that are used for determining which patients should be entered into trials or whether the patients responded to therapy. And this could be a huge advance for standardizing trials and ensuring that if you're comparing different trials, that you're really focusing on differences in drug effect, not differences in pathologist scoring. And just to get some sense on reproducibility of the results using the AI system, the graph on the left, where the blue is the algorithm output and the gray is manual scoring, is just the algorithm alone in blue, whereas the one in the middle is the algorithm supporting pathologists. And you can see you actually get almost identical results, whether you're comparing algorithm alone versus ground truth or algorithm assist versus ground truth, which again should give us a lot of confidence that this will make pathology a much more robust field that is not so dependent on the few experts, but really can democratize expertise through deployment of models like this. So I wanted to use that as kind of an intro to talk about really the focus of today's session, which is HER2. We built a very similar tool called the AIM-HER2 algorithm that takes as input an IHC stained HER2 slide and automatically identifies artifact, invasive cancer, and then produces quantitative and categorical HER2 scores. And to take you through this figure for how these systems work, the image on the left is the standard of care, a whole site image of HER2 IHC. Then an algorithm is run on this image to identify all artifacts in yellow. So it makes that a highly reproducible just quality assessment metric that we can now get on every single slide from every single lab using the system. And then the third set of models are applied to identify, in this case, epithelium, cancer epithelium in red and cancer stroma in green. And then fourth is this HER2 prediction overlay that goes on top, where this additive multiple instance learning model has classified every region of invasive cancer into HER2 0, HER2 1+, HER2 2+, or HER2 3+, and then there's also a slide level summary algorithm predicted HER2 score for the whole slide. So now for every case, in an automated fashion, even before the pathologist sits down, they have these quantitative metrics for the case, and each of those could be used in the research setting to correlate with response, and then the algorithm predicted HER2 score could potentially assist a pathologist in determining their own score. So we wanted to conduct a prospective clinical trial, very similar to what we did in NASH, and we actually just completed this past month, where we had a reference set of 5 pathologists, not using AI as our ground truth, and we obtained consensus HER2 scores from the panel of 5 pathologists, and then we had a total of 20 pathologists, and it was a crossover study design, where cohort A was 10 pathologists, cohort B was 10 pathologists, and each cohort did either AI-assisted HER2 scores, indicated in purple, and then had a 3-week washout period where they then did the opposite arm. They either did the manual if they started with AI-assisted, or AI-assisted if they started with manual. And then we also had an arm where we just ran the algorithm by itself. So we haven't actually seen many others in the HER2 setting. I'm not sure any of the other algorithms have been validated in this way with a prospective crossover study design. But I think the key results to focus on are here, where AI-assisted HER2 scoring is superior to manual scoring across HER2 levels. We see the overall agreement on the left, and this is agreement of AI assisting pathologists to the manual consensus ground truth. So this is really the accuracy side of error, not even the reproducibility side. We know reproducibility is going to be far better, but here it's showing it's even more accurate as well. So left is lumping all the groups together. I think the one that's most relevant for this talk is probably D, which is just focusing on the 0 versus 1+ distinction, where we see significant improvement with AI assist versus manual scoring. And I think the beauty of it is that it's globally deployable, so it can really be applying the same algorithm to everyone, which would greatly standardize the assessment of this distinction across the world. And so we've been excited about the HER2 results and ERPRQ67 and have built these algorithms into this RUO platform called AI Site so that researchers or pathologists can deploy these on their breast H&E, and in this case IHC slides, to provide in a fully automated fashion algorithms that identify artifact, identify invasive cancer, in situ cancer, and quantify the key biomarkers in breast cancer. And I think this is really where the field is going, and algorithms have been available for some time, but I think this clinical integration into a platform, integration into workflow has been one of the things that has lagged behind that we're finally catching up to. So that's one, I think, super important part of the picture. I tend to think it's the most important part because I think IHC is a powerful technology, and it's the reading of it that has left so many opportunities for improvement. But it's not the only part of the equation, and I do think in parallel with improvements in interpretation, which I think will be driven entirely by AI and AI assist devices, there's definitely room for improvement on the assay side. And David already mentioned this, I believe, but his group published a really nice study earlier this year showing that they can build a highly sensitive platform for quantitative assessment of, in this case, HER2 to measure at much lower levels accurate quantities of HER2 at the attomolar per square millimeter. So to be super precise and accurate in the measurement. I think given the growth of immuno-oncology, of ADCs, of how difficult it is to get biopsies from patients, there's going to be a huge need for multiplexing and really effective multiplex technologies. And you think scoring one stain is hard. Scoring 2 or 3 is basically impossible to do at any kind of scale reproducibly. So in parallel, AI-based measurements, I think, are going to be really important for building along with novel high-sensitivity assays. I think over time, more and more of those will be multiplex, not uniplex, and I think they'll enable more accurate, reproducible, and highly sensitive measurements of ADC targets, because I think there is a future where it's not only are you positive or negative, it's really which of 3 or 4 therapeutic options are you the best candidate for. And there you really need reproducible ways of comparing different markers. And that requires highly accurate and reproducible analysis. So far I've talked mostly about using AI and quantification to kind of measure what we already know is important, but just measure it better. So like when you look at the HER2 outputs in our AIM HER2 score, they're the standard ASCO CAP categories and then the standard slide level biomarker. And I think there's a ton of power for improving the accuracy and reproducibility for biomarkers that are important. But the beauty of digital pathology and applying AI to digital pathology is on the exact same slide, you can measure the biomarkers that we already know are important, but do a lot of discovery-based analysis to discover novel, in this case, immunohistochemistry-based phenotypes that could be predictive of ADC treatment benefit. AstraZeneca has been a real leader in this field. They've developed a system called quantitative continuous scoring. They published a small study earlier this year on one small data set in Scientific Reports for their work on HER2. But essentially, they're taking a machine learning approach to discover novel quantitative phenotypes to find new biomarkers beyond the standard readouts that are most predictive of treatment benefit. And here they show in their schematic that they build sort of the image processing side on the left and then lock the models, and then they build models optimized to survival on the right and then lock the models, and then validating these locked models on unseen prospective data will be a really important next step. We've done similar work at PathAI. This was a study we published or presented at ESMO in 2020 about an AI-based analysis of advanced breast cancer patients from a phase 1 trial of T-DXd to identify novel features predictive of prognosis in this single-arm study. And we identified novel features of the tumor and the tumor immune microenvironment that outperformed either standard biomarkers by AI or certainly manual scoring. And I think that is the future, where we have the standard readouts from the tumor, but potentially novel readouts, in particular from the tumor microenvironment, including the fibrotic microenvironment and the immune microenvironment, that will combine to likely be the most predictive biomarkers. And you can get all that information from the exact same images. So the last schematic, just to really show you in practice what this looks like to go from a normal biomarker, standard biomarker, to a novel biomarker. This whole population of patients on the left could all be grouped as HER2 3+, based on standard ASCO cap categories. And you really don't see much difference between these two patients, the orange one and the blue one, based on average membrane intensity. So just taking the average across the 100,000 cells in each patient doesn't really tell you that much that they're very different. But if you apply a tool like IHC Explore that can give you single cell resolution readouts, you can actually see that there's very different distribution of cells positivity within those two cases, where the blue case is sort of a unimodal distribution that really is has an average membrane intensity that's representative of the distribution, whereas the yellow case is actually a bimodal distribution that has totally negative regions and then very positive regions. And we really have no idea how this predicts response to therapy or recurrence risk. And now with tools like IHC Explore, we can begin to discover novel spatial patterns of ADC target expression that may be most predictive of treatment benefit. And I just wanted to end with the importance of collaboration with folks who can really take this all the way through, from translational research to validation, commercialization, and clinical distribution. And I think this really is where this field is going, is not only used on the left, but to really be able to take these all the way through to new CDXs. And we formed a partnership with Roche Tissue Diagnostics, one of the leaders in CDX, to enable the development and regulatory approval and commercialization of this future of AI-powered CDXs. So I just wanted to end, I know I'm a few minutes over. I think AI will be an important part of the solution to the pathologist conundrum. Establishing the accuracy, precision, and clinical validity of these AI-based tools will be critically important, and these efforts are already advancing rapidly across areas, oncology, MASH, IBD. And this last point, I think, can't be overstated, that these collaborations are so critical between AI companies, the assay manufacturers, assay developers, drug developers, regulators, payers, and most importantly, physicians and patients, to really make these not just exciting tools that are used by a select few, but that they can really positively impact the way pathologists and oncologists practice, and ultimately can really bring benefits to patients. So thank you.
Okay, great. Well, there's 2 microphones, and so anyone that wants to come up to the microphone can ask questions. And I also have some questions that we've gotten online that we'll address. But since you came up to the microphone, you can be first.
Thank you. Judy Ann Chapman. I declare a biostatistician who has worked for more than 4 decades with dear biochemists and pathologists who have explained the great complexity that you have all been talking about. So I really understand you, and thank you. One of the comments, I've got a different approach to what's being suggested today, and that's an adjunctive after your good lab work, your best quality control. I don't think we can do without it. But it's statistical standardization. And I've worked with quality control labs, including I did some time with UK NECOS as well. And what we've recently shown and published in JCO is that ER and PGR, statistically standardized, provided more clinical information than ASCO cap. Now, I've been working very recently, it's a late-breaking abstract here, on HER2. And you can find the abstract and the poster. We think we have some more information as well after best lab, that the statistical standardization, and I should say for the AI person, I'm fully behind you. We've been using quantitative image analysis as well as all the other best lab processes. So I just offer you that there is another thing that I think will help. And I'm really appreciative that the pathologists are getting the word out that we need it. Because I think it's been a communication gap between the laboratory people and the clinicians, and you're helping to overcome that. Thank you.
Thank you. Dr. Salgado.
Yes, thank you very much. Roberto Salgado, Antwerp, Belgium. I will start with a disclaimer. I'm a firm believer of the added value of AI in the community of pathologists, but I have a more cautious approach. We presented, and that's my first question to Andy, and I have a second one to Beppe, and maybe a third one to David. We presented at this meeting a comparison between 10 different AI tools for HER2. And we show that at the high end, it's pretty good comparison, but at the low end, the concordance between these tools is actually worse than between pathologists. And so I'm more nuanced about the term accuracy, reproducibility. When you compare 10 different AI tools for HER2 and find more discordances than between pathologists, according to the AQA data. So and you can approach it in two different ways. So at this conference, I heard about AI is Lucius because it's worse than pathology, so why should we use it? But the more constructive approach is, and that's my question to you, how can we improve the concordance between these different AI tools? So that's the first question. Related to that, and that's a question to Beppe on his comment on prediction. That's more important than being more precise. What would be a solution to solve this issue? Would it be that, for example, in the 04 and the 06 study, that we get some way to compare these slides and apply those 10 different AI tools on those slides to see whether it matters clinically or not? Would like to hear your opinion on it. And wait. And then the third question, that's for Kim. Let's imagine that we would be able to apply those 10 different AI tools in the 04 and the 06 destiny studies, and that it doesn't matter at all whether it's 0, ultra low, low, 1+, or whatever. Would it impact the next ASCO cap classification of HER2? Thank you.
Andy, you go first. Yeah, I think so. Push your button, I think.
Sorry. It's great work that the Friends of Cancer Research are doing, and I think that was an important poster. I think it's truly a hard problem. I mean, I also think this AI assist concept is important in the same way that pathologists are different. The same algorithm will give you the same result every time, but different algorithms. There's no guarantee at all that they'll give you the same result, like zero guarantee. And to some degree, the fact that they're so reproducible hurts, doesn't help. So it's like when you're wrong, you're just going to keep being wrong, you know? Versus, say, a human where your judgment, maybe you oscillate around what's accurate. But algorithms, if they're right, it's great that they're super reproducible. And if they're wrong, it's very bad that they're super reproducible. So it's a real problem. What are the solutions? One, I think having both analytic and clinical validation is obviously very important. So how are these impacting clinical behavior? I don't think there's an obvious solution other than having like a ground truth that everyone agrees on, that's standardized. I think that's a difficult thing to do, but other fields in computer vision have done that, where they just have like 1,000 images that are held out that some independent body determines performance against, and maybe there's a training set that's common to all the different companies. I do think that would work, because you have to actually optimize to something, and given how the ground truths are so variable, one thing we show in this study that I didn't show is the 5 pathologists that made up the panel would have a big impact on what the ground truth was. So we had a total of 25 pathologists, and we had 5 who were part of the reference panel, but just as an experiment, we did all these selection of random sets of 5, and the ground truth moves around a lot. So I think having an agreed upon ground truth could be really important. And of course, having clinical data at the end of the day. So having clinical trials to deploy this on and clinical validation. The other thing is it's very, very easy these days to build an algorithm. It's very hard to build a whole platform that integrates into pathologists, that potentially has regulatory approval. And I think separating those two things out is really important, and educating pathologists about that. What's an RUO? What's got FDA clearance? What's integrated into a platform? I mean, and that will at least narrow it down.
Yeah, shortly, Roberto. What I'm saying is that sensitivity not always compares with prediction. We had run a bridging study for the Impassion 130 study that was based on the SP142, and we have retested the vast majority of the tumor samples with the 22C3. 22C3 stained 40% more or something like that cases, but those additional cases, apparently they did not receive any benefit from atezolizumab. So my sense is that in order to have at least a sense of the predictive value of this alternative assay, we have to run bridging studies, or alternatively, in the design of the clinical study, you have to foresee that not all the world will be using that test for that reason. And so maybe as a stratification factor, allow people to use whatever test they want, and then eventually at the end of the trial, you will say which is the test that is more predictive.
Yeah, I'd second that thought. I really like that idea. I think clinical trials need to integrate those questions. I mean, we can do them retrospectively. It becomes a lot harder to move the needle or to do bridging studies. To your question, I mean, I can see if you found the perfect tool, here's the AI that is correct and predictive, then your question is access, right? So, you have to recognize that not everybody has digital pathology. The majority don't. But we need to move towards that, and maybe it'll be just reference labs that have it that you do your test, at. But that'll be an access issue. And then to answer your question on would we change guidelines if AI couldn't find a predictive? I mean, we already kind of have a signal that 1+ versus ultra low versus 2+, the level of expression didn't seem to matter in terms of response, right? They all responded, at least that was the data, and it was a mix of 1+, 2+ that responded.
I don't know whether we should focus on the magnitude response, because if we continue to derive from the clinical trials the positive versus negative or ITT population, it will not help us. I think that we desperately need to get access to the granularity of the data.
Let's let the other author from the ASCO CAP guidelines speak. Do you want to ask your question or also put a comment in, Antonio?
Yeah, I do. I have a comment and two quick questions. So, Antonio Wolf, I'm a medical oncologist. I played pathology on TV, but I'm not a real one. Just a quick comment. I think I'm speaking for myself, I think we failed you pathologists. Raise your hand how many pathologists are here. There are a bunch of you. We have put you in an impossible situation because of a label, pure from an FDA label, for me to be able to give trastuzumab deruxtecan to my patient, I need to give you a box of chocolates for you to say this tumor is not IHC 0, this tumor is IHC 1+. And I am yet to hear a reason why the companies, the sponsors, made a decision of exclude patients that were IHC 0 and more recently IHC 0, no staining from the clinical trials. Because if that had happened, we would have data on it. And at the end of the day, this could be a gigantic exercise in futility. We are trying splitting the zeros. It's really crazy. And so we need, right now, we have no evidence that this is a distinct biological entity to justify a new classification. Dr. Beck, I have a question for you. You showed some very interesting data. I'm a firm believer in all that you showed today. You have not, and you are collaborating with Roche, you may be collaborating with AstraZeneca, Daiichi perhaps. Are there plans to apply some of your studies to the existing data we have to see if we can identify a differential benefit or differential response to trastuzumab deruxtecan in the clinical trials according to potential different levels of HER2. And my one concern is that IHC was never developed for this. It was developed for high levels. It has a wonderful dynamic range for high levels, awful for low levels. So is it going to be possible to test this?
Yeah, it's a good question. Just kind of sharing what's in the public domain. We showed one poster from 4 years ago with Daiichi on J101 where we did find this novel biomarker and were able to stratify better than manual scoring. They just published this paper, AZ did, on actually the same data set just this past year, using their, like as a one demonstration of their QCS system where they did aim to better stratify and show that they could find HER2 negative patients who respond using their system. What do I think? I don't know if people are going to try to refine HER2 or, I feel like there was a ton of work that happened on PD-L1 after it was already out and the CDXs were already approved. To Kim's point, I think it's hard to do these things after the fact, and it's much, much easier to do it prospectively. So what we're doing with Roche, we just announced a big partnership earlier this year to really try to be involved very, very early in assay development, to develop the assay along with the AI, to do exploratory research together, and then ultimately to design trials. So I think the prospective nature is really important. And once a CDX is on market, I think in general you don't see big changes beyond scoring assist tools or sometimes, but big changes in like criteria.
And I will never suggest that we could have done any better, but I must admit that I am disappointed that the companies did not approach any one of us thinking about, who knew a little bit more about immunohistochemistry when they were making decisions about launching the study the way they did. Dr. Philip, I have a question for you because we, and I don't know the process, how it took place. The FDA extended the label of the assay to, and used the company terminology of IHC low. This is a company-created terminology. There are no data to support that. And I'm curious about what's going to happen now because we have a very exploratory subset of DB06 with 72 patients in each arm that were called by the company IHC HER2 ultra low, which actually happens to be the IHC 0, a little bit of staining, some staining from the ASCO cap. One could argue that an exploratory objective should never be enough for the FDA to extend the label to the drug. That's the purest view. On the other hand, from everything we know, there is no evidence that there is based on IHC, that there is any way to identify someone who's unlikely to benefit. So my sense is, at this point, everybody should be given the drug. Roche, I don't know if they want to find out whether there's a differential level of response because they want to have as broad of a market as possible rather than shorten the market. So my question to you, Dr. Philip, is whether if the company is coming or will come to you, instead of putting out a label, HER2 ultra low, on the basis of 140 exploratory studies or samples, just use what ASCO cap did and call it IHC 0, some staining, IHC 0, no staining, and diminish the confusion for the pathologists. But until then, you guys are stuck because I'm not aware of any prospective randomized trial testing TDXD in IHC 0 no staining, whether it works or not. That study is not going to happen, and I'm fearful that we are stuck.
TB15.
So,
IHC 0 trial. Yeah. Do you want to answer, Dr. Philip?
Yeah, I mean, good point. It's something we could actually discuss because, as you very well said, what sponsors bring forth is what FDA looks at. So if they propose that terminology, there are sometimes we do say this is not correct. For example, if somebody's changing the antibody but still keeping the same name of the the test, we tell them no, you can't do it because then there's a confusion. The other one is already out there, so this one has to be changed. Otherwise, there is no way people will distinguish between the two, because unless the other one is withdrawn from the market. So something like that, it's very clearly said. But this one, yes, clearly they said it's a low, because unless we say 1 + 2 + 3 +, so it was a terminology proposed by them.
Okay, thank you. We're actually 4 minutes over, but there's no one chasing us out, so if people need to leave, they can do so, but I don't want to miss taking a question from Dr. Bloom, who was one of the pioneers in this space.
So I have so many questions and so many comments, I could go on for an hour, but I'm going to make it easier. So very beginning of HER2, right, we had other studies. We had radioligand binding assays that showed us distribution of HER2 by cell. We had EIA assays that showed us continuous, just like the old ERPR, grind and bind assays, but gave us really nice linear continuous variables. And what we saw was that HER2 was distributed in a nice bimodal distribution. You had a nice peak, you had a nice trough, and then you had a nice peak. So the 3+ was easy. You could take that end peak and you could say, this is 3+, and the trough corresponded to the 2+, and that meant that some of the tail end of the 3+ was in the 2+. The 1+ and zeros were never distinguished even by EIA, right? It's a one big curve. And when you take that tail end at the 2.5%, 5%, whatever that tail end of the curve was, that's a large number of cases. And in fact, what the radioligand binding showed was that 5% of cases underexpressed HER2 relative to normal breast epithelium, which, by the way, isn't zero, it expresses 20,000 receptors. So we know that all epithelial cells express HER2 at some level. We know no matter what 15 shows, Destiny 15 winds up showing, that 5% of the zeros are probably true underexpressors even to the normal cells that are around. And they might be such a small fraction relative to the other zeros, because again, one big peak with a huge curve, we just put a line, I don't even know where the line is for the assay, right? Because nobody did reproducibility. So it's like, yeah, when we saw something a zero, it's not a zero. And I'm not sure, and the real question, as David, you pointed out, we've had many discussions on, the assay that we have, the IHC assay that we have, and as Antonio pointed out, it's not linear at that range. So trying to distinguish where we're at in a curve, we know what the curve looks like. We know what the receptor distribution looks like, right? There's no way to draw an arbitrary line. The other thing to notice is if you do receptor counts versus the standard 0, 1+, 2+, 3+, it goes roughly 20,000, 100,000, 500,000, 2.5 million, right? So the curve, if you plotted it, isn't a 0, 1, 2, 3 that looks like nice linear and we say, oh, we'll just move the cut here. The curve is like this, right? And so you just move a fraction over and you're not talking about a small number of cases in that differential, you're talking about a huge number of cases in the differential. To Andy's points, we've done computer vision. The continuous scoring I laugh at because that was the old ChromaVision from 1999. That's what it did. It did a densitometry score. Go score and it'll quantify the amount of brown over blue. We do it more sophisticated now, but that was the old system. And that data, of course, showed that pathologists are more reproducible using the system. Because your eye can't see that color difference, the machine can. So we can get a reproducibility issue, but I'm going to go to Pepe's point. What matters is the predictive power. And the assay that we're using isn't fit for purpose for distinguishing a curve that looks like the biology. So we need to go to something like you're developing, either molecules, attomoles per area.
I would just comment on that the
Right.
attomoles per square millimeter exactly agrees with what you said, that
Exactly.
5% of the cases are below the limit of detection.
Exactly. I mean, the science is the science, right? We can't change the biology. Those studies were done, by the way, before Herceptin. Those were HER2 studies when we were trying to understand HER2 and how it was distributed in breast cancer. So it is unbiased, uninfluenced by any therapies because they didn't exist yet. I don't think we could throw those old studies out. We have to see what we're doing and how it correlates. But to Pepe's point, what we do as pathologists is interesting, but what's more important is can we predict behavior of the drugs?
Thank you. Thanks, Ken. So there's 10 questions up here, and I will we'll stay here and answer questions out of individuals. I can't go through these all. We're already 8 minutes over, so thank you all for attending, and let's thank our panelists.
Thank you.